Hello everyone, I'm Dr. Tahir and welcome to my usmle Step 1 lecture. Today I'm going to talk about Lac Operon. The approach that we will have is I'm going to first read out the question for you all, and then we will go through the explanation, and finally at the end I will give the explanation of each and every option. So here the question says, a microbiologist is investigating sugar metabolism in a non-mutant E. coli colonies grown in a glucose-lactose medium. The medium undergoes Western blotting technique using a labeled fluorescent probe for beta-galactosidase and permease. Despite of having lactose in the medium, only trace molecules of beta-galactosidase and permease were found. Which of the following explains the finding? A. Increased binding of lactose to repressor. B. Increased cyclic AMP-cap interaction. C. RNA polymerase cannot bind to promoter. D. Decreased catalysis of ATP substrate. E. Increased cyclic AMP to ATP ratio. F. Operator locus is not free. So talking about Lac Operon. So what an operon is? Operon comprises of the group of genes which are regulating other genes for the expression of synthesis of proteins. So i.e., when you talk about the operon, there is a group of genes which are regulating the expression of other genes for protein synthesis. And that is for lactose metabolism. What I'm going to do first is, here we can remember the mnemonic, and the mnemonic is CIPROG. CIPROG, C stands for catabolite activator protein. I is lac I. P is for promoter. R is your repressor. O is operator. And G is for genes. The cap, catabolite activator protein. Catabolite activator protein. Name itself says catabolism. That means this protein is responsible, this catabolite activator protein, it is basically responsible for activation or inducing the gene expression. Now how it is going to do its job, I'm going to mention in a while. Like it's an inhibitor. Inhibitor means what? Basically it is responsible for the synthesis of a repressor protein. Synthesis of a repressor protein which will bind to the operator. I'm going to give the picture so that it will be much clearer. Promoter, as we know in the eukaryotes as well, that's the site where RNA polymerase binds. So it's an RNA polymerase binding site. RNA polymerase binding site. Repressor. Repressor is, it binds to operator in a way it negatively regulates. Now I'm going to mention about negative and positive regulation. Negatively regulates the gene expression. So that means if I don't want the gene expression, if the E. coli doesn't want the gene expression, then the repressor will be bound to operator. Operator is the site where repressor binds. Some books refer operator as an on-off switch. It's an on-off switch. The genes. These are structural genes. Structural genes means what? Basically, these are the ones whose expression is necessary for the synthesis of enzymes to break down lactose. Now, what are those genes? Lac Z, Lac Y, and Lac A. Individually, we will discuss about this in a while. So, catabolite activator protein as we see here, the negative regulation is by repressor. The positive regulation is by cyclic AMP binding to cap. So cap-camp interaction. This is positive regulation for gene expression. Having said that, I'm gonna write down and draw the picture for you all for the unit of genes, that is your operon. So the unit of gene, the operon, the picture looks something like this. This is a site for cap, for a promoter, operator. This is for like Z, Y, and A. This is the site for Lac I. I'm not going to draw the promoter for Lac I, which is low yield. Here is your catabolite activator protein, cap site. This is the promoter, this is operator, this is Z, Y, and A. Where is the repressor? A repressor protein is going to bind somewhere here. Now you see, I just now mentioned that this repressor protein synthesis is by the inhibitor, that is Lac I. Lac I stands for inhibitor, which is taking part in the synthesis of the repressor protein. I told you repressor was positive or negative regulator. It was the negative regulator, right? Now the positive regulator was catabolite activator protein, which is bound by cyclic AMP. So here what will happen, first of all, I have to just make it clear in front of you that the operon which is there, it requires two important functions. Repeating first of all what I said, that is cap-camp interaction. Cap-camp interaction, this is the positive regulation, and the negative regulation is by a repressor bound to operator. Repressor bound to operator. Repressor bound to operator. What will happen if repressor is bound to operator? We know in eukaryotes, promoter, this is the site where your RNA polymerase comes and binds, and then gene expression occurs. So these structural genes like Z, Y, and A, the expression of gene occurs when the polymerase will come and bind. When the repressor is bound to operator, RNA polymerase cannot bind to promoter. The repressor is bound to operator, RNA polymerase cannot bind to operator. So repressor is bound to operator, RNA polymerase cannot bind to the promoter, and there is no gene expression. So there should be one R. You want to remember like this? There should be one R. Either repressor should be there or RNA polymerase should be there. Now, here RNA polymerase cannot bind to promoter. Not bind to promoter. Right? Coming back to, if you understand this cyclic AMP, where the cyclic AMP comes from? You might be studying in your by the glycolysis. And what is that? That if glycolysis occurs, what is the intention for glycolysis? We need ATP. Like in eukaryotes also, the sequence of steps, I'm not going to discuss the glucose to glucose 6-phosphate, glucose 6-phosphate to fructose, fructose 6-phosphate until you go to pyruvate. What is the role? Well, the main intention is to get the ATP. Or you get one NADH that will enter the electron transport chain and you will get 2.5 ADP. But ultimate intention is to get the ATP. For the survival of the bacterium, it requires ATP. And for that, the best energy source for E. coli is glucose. So glucose being one of the most important energy source, so if you will see, If glucose is present, that's the best source of energy for E. coli. Best source of energy for E. coli. Whereas, if glucose is absent and lactose is present, then the Lac Operon is active. So Lac Operon is active in this state. Keep in mind, no glucose, lactose must be present. How it will regulate it? It will be regulated in a very simple way. If glucose is present, then immediately this glucose will inhibit an enzyme, that is your adenylate cyclase. Adenylate cyclase. So glucose will inhibit the enzyme adenylate cyclase. Now, let's say that there is no glucose, which we want to study here for Lac Operon. That means whatever the ATP is there, it is decreasing or depleting. That means adenosine triphosphate. We know that when ATP decreases, it becomes ADP, and then adenosine diphosphate, then adenosine monophosphate. So for here, ATP will convert to cyclic adenosine monophosphate by enzyme adenylate cyclase. Simple. It's really simple. Adenylate cyclase. So adenylate cyclase enzyme converts ATP to cyclic AMP. That means you are decreasing, or E. coli is having depletion of ATP, and cyclic AMP concentration is rising. And this cyclic AMP, it binds to catabolite activator protein. And I told you this was binding would lead to positive regulation. That was positive regulation means camp-cap interaction was a positive regulation for gene expression. But think about it. Even if it binds to cap side, if there is a repressor bound, I told you there is something that is negatively regulating. That means there is no gene expression. Still, because of this repressor bound to operator, there is no gene expression continued. So point to be understood is, what is the reason here? Because of this, you can understand that operator, which is the site where the binding side of the operator is by the repressor. And remember, this operator is in off state. Operator is in off state. When operator is in off state, there is no gene expression continuation. And you see in Lac Operon is for lactose metabolism, breakdown of lactose. It says that lactose breakdown will occur when the operator is in on state. So operator must be in an on state, then only the lactose metabolism can occur. So point, what will happen here? Simple thing that's going to happen is, I told you that there should be lactose in the medium present. There should be lactose in the medium present, then what will happen? Lactose comes and binds to this site. First, in the beginning, once this lactose comes and binds to the site of the repressor, this repressor is unbound. As the repressor unbounds, then RNA polymerase can bind to the promoter site. And that induces the gene expression for these enzymes. What enzymes? The proteins, the enzymes, the proteins, the enzymes, the proteins, LAC-Z, LAC-Y, and LAC-A. So these three genes encodes for proteins like beta-galactosidase, Y stands for permease, and LAC-A stands for transacetylase. So three genes, three proteins which are synthesized. The three proteins, beta-galactosidase, permease, and transacetylase. Beta-galactosidase has got the role just what in eukaryotes lactase has got the role. So what lactase used to do, we know that lactase, just like in eukaryotes we were having, we know that lactose is converted to glucose and galactose by enzyme lactase, right? Lactase. And then this glucose becomes here E. coli gets the glucose as the principal source of energy. As I said, in eukaryotes, if you see, like galactose will convert it to galactose 1-phosphate, galactose 1-phosphate will convert to glucose 1-phosphate, and glucose 1-phosphate will convert to glucose 6-phosphate, and you can see glucose 6-phosphate is one of the steps in your glycolysis. By galactokinase enzyme here, galactose 1-phosphate uridyl transferase, which is defective in classic galactosemia. Now, I'm not here to discuss about the biochemical steps here, but to understand is what lactase is doing is the same job done by beta-galactosidase. But you see here the simple thing, what E. coli is getting, the principal source of energy is glucose. That means glucose starts rising. Still, despite of having galactose, glucose becomes the energy source in E. coli. Once this glucose is present, after breaking down, then this glucose can inhibit the enzyme adenylate cyclase. So glucose can again inhibit this enzyme. That means when you are having enough ATP, there is no need of cyclic adenosine monophosphate. There is no activation of this enzyme. That means you are having enough ATP. So point to understand is that beta-galactosidase enzyme is responsible for doing the job what lactase enzyme does in eukaryotes. One more important point here, that why this all steps are active in the E. coli. The reason is because bacteria doesn't like to waste the resources. So this Lac Operon should be active only when there is a need. When you are literally having no glucose present in the medium, then only the Lac Operon is active. The next important thing is the steps that we are discussing here, like in eukaryotes we see transcription and then translation, the steps that occur, like transcription inside the nucleus, the DNA to mRNA, and then it is capped and tailed, then mRNA leaves the nucleus, enters into the cytoplasm, either it is stored in the P bodies or later it can take part in the translation, right? And finally the proteins will be synthesized. But here in E. coli, there is synchronous. There is a synchronous transcription and translation. One very important point as far as your USMLE is concerned, it is there in multiple different Q-banks also. You should remember about the synchronous. Another very important point to remember is, as you see here, there are different proteins which are synthesized, right? There are various different genes here. In eukaryotes, we have specific gene for a specific protein controlled by a specific promoter. But here in the operon, you see that there is a one promoter, but more than one genes. Such an mRNA, which has more than one genes, synthesizing more than one proteins by a single promoter, is a polycistronic mRNA. Is a polycistronic, poly means more than one. In eukaryotes, it's rare to find a polycistronic mRNA. What is the role of this particular other enzymes like permease? Permease is responsible, just like, like E. coli. When you study about the permeability, like you're bringing in more lactose. So permease brings in more lactose inside the E. coli cell. And transacetylase, its role is not very well clear, but it says that transacetylase is responsible for transfer of acetyl group for beta-galactosidase enzyme. Transacetylase is responsible for transfer of acetyl group for beta-galactosidase. So that means once this lactose is finished, then what will happen? Once this lactose is finished, we know that before this lactose was binding to the repressor, and then it was creating a conformational change, and then repressor was unbound. But once this lactose is used up and there is no more lactose bound to repressor, the repressor goes back and attaches to the operator. And in a way, again it starts negatively regulating. And that should do for the Lac Operon. So what we understood in the question is that there is a medium where there is a glucose as well as lactose present, and that the medium was undergoing Western blotting technique. Western blotting is for recognition of proteins. What are the proteins here? These are your proteins. And then you recognize these proteins and you check for the availability of the proteins. And then you use a fluorescent label probe to recognize whether this protein is present in the medium or not. And then what you saw here, that despite of having lactose in the medium, there were trace molecules of beta-galactosidase and permease. In other words, the gene expression was not occurring. And why gene expression was not occurring? Because glucose was present in the medium. And if glucose was present in the medium, what was not happening? Glucose was inhibiting the enzyme adenylate cyclase. So adenylate cyclase enzyme when inhibited, no cyclic AMP. No cyclic AMP, that means the ATP concentration was high and cyclic AMP concentration was low. In other words, ATP to cyclic AMP ratio, ATP to cyclic AMP ratio was increasing. There is an increased binding of lactose to the repressor. It doesn't explain the situation. Here the situation is explaining clearly that there is no positive regulation, and the question is talking about the negative regulation, that is repressor lactose binding to the repressor. So here this is not at all an answer. The next, increased cyclic AMP to cap interaction. There is actually decrease in cyclic AMP cap interaction because of adenylate cyclase enzyme inhibited by glucose. So again, this is not the answer. RNA polymerase cannot bind to promoter. This again doesn't explain. What is the problem here? That is explaining why there is trace molecules of beta-galactosidase and permease. It is because of glucose inhibiting adenylate cyclase. So you are not having CAMP-CAP interaction. That should be the explanation. You are not having enough cyclic AMP. You are not having ATP to cyclic AMP conversion. So this doesn't explain. This is just like explaining indirectly option A, because in option A also there is an increased binding of lactose to the repressor. Even if lactose binds to the repressor and let's say the repressor detaches, if RNA polymerase is coming and binding to the promoter, there is no positive regulation and further gene expression because there is no CAMP-CAP interaction. This again is ruled out. The next option is here, I will go with the E, increased cyclic AMP to ATP ratio. Increased cyclic AMP to ATP ratio, here as I said, there is a decreased cyclic AMP to ATP ratio in this situation. Why? Because adenylate cyclase enzyme is inhibited. So again, this is the other way around. There is an increase in ATP to cyclic AMP ratio because ATP concentration is high. This is not the answer. Option F says operator locus is not free. Even if operator locus is not free, that means what? We know this is saying that operator is in off state. Still, it doesn't explain why adenylate cyclase enzyme inhibition by glucose is leading to the trace molecules of these enzymes. Doesn't explain. Operator locus is not free. That means the area is bound by a repressor. It doesn't explain. What D option says that there is a decreased catalysis of ATP substrate, and that's true. Isn't it that there is a decreased conversion of ATP to cyclic AMP? So when there is a decreased conversion of ATP to cyclic AMP, there is a decreased catalysis of ATP substrate. And this is the answer here, that is decreased conversion of ATP to cyclic AMP because adenylate cyclase enzyme is inhibited by glucose. And that's the end of our discussion. Thank you.